I would like to welcome everyone and thank you for joining us for the ICR webinar from the month of May. My name is Chad Kinney. I'm the director of the Institute of Cannabis Research, and I will be moderating today's webinar. Before we get started with the presentations, I'd like to discuss a few housekeeping items. Today's presentations will be followed by a short question and answer period. To ask a question, we ask that you use the Q&A function in Zoom. You can enter a question at any time during the presentations. We will address as many questions as time allows up until 2 p.m. local time. For any technical issues, please use the chat function in Zoom. Both the Q&A and the chat functions should be found in the toolbar on the Zoom screen. This webinar series is an opportunity for the ICR to facilitate sharing of information related to cannabis research, whether that is about the research itself or mechanisms to facilitate research, as in the case of the presentations today. The webinar series offers an opportunity to go in depth into these topics. The ICR also hosts an annual research conference in partnership with our colleagues at the Global Hemp Innovation Center at Oregon State University. This year, the conference will be virtual and occur the first week of August. The conference offers an opportunity for participants to engage in a wide range of, or engage with a wide range of researchers. This year's conference will have well over 100 presentations and panels. If you're interested, you can certainly find out more information about the conference on the ICR's website. And we will also post a link to the conference website in the chat window while the presentations are going on. This month, we have the pleasure to hear from two presenters, Mr. George Hodgin and Dr. AJ Nayak. Mr. Hodgin will be delivering the first presentation today. He is the founder and CEO of Biopharmaceutical Research Company, or BRC, which is an applicant with the Drug Enforcement Administration to produce federally compliant cannabis products for research purposes. BRC holds several DEA Schedule I registrations and engages in compliant cannabis research. He is a former Navy SEAL officer and led SEALs in combat and counter-terrorism operations in Afghanistan and Southeast Asia. Mr. Hodgin earned degrees in Economics and Mandarin from the University of North Carolina at Chapel Hill and is an alum of the Stanford University Graduate School of Business. With that introduction, I will turn it over to Mr. Hodgin to give our first presentation of the day. Please feel free to turn on your camera if you like.
Thank you so much, everyone. Thanks for tuning in, and a special thank you to Dr. Nayak, my co-presenter later in the session, and certainly to the ICR for hosting not just this webinar, but for this really wonderful series that they do, and for their newsletter as well. We're certainly proud to be a part of it. What I want you to take away from the presentation today is that federally compliant cannabis research is hard, but doable. If you go through the appropriate processes, it is doable, although it might seem complex and intimidating at first. So I'm going to talk a little bit about what that process is like to become federally compliant from a DEA registrant perspective, as well from an industry perspective, which will dovetail into Dr. Nayak's presentation around collaboration and partnerships. Before we get started, I'll give you just a quick background that'll highlight, I think, the need for cannabis research and why what the ICR is doing is so important. I got involved in the cannabis research space while I was a student at Stanford, as Chad alluded to, and I got involved in the healthcare of a very close friend of mine who was a fellow Navy Special Operations veteran. And when I was at the VA, the Veterans Affairs Hospital, helping my friend and former teammate with their healthcare, he was going through all sorts of opioid abuse as well as PTSD, and he said to his doctor, "Hey, look, doctor, I'm going to try medical cannabis and see what the buzz is about. Could you just tell me where to access it and how much I should use?" And this physician responded to my friend, "Look, at the end of the day, all the stuff that's going around in the States shows us it's not going to kill you, but But for me as a physician to answer those questions is really challenging because there's an incredible gap and need for meaningful scientific research around cannabis. That's why we decided to dive right in. Next slide, please. So as we, there we are. So as you all in the research community are all too formally aware, it's really challenging if you are an academic or a biotech scientist, someone that's regulated by federal law. The discrepancy between what goes on with consumers at the state level and states that have legalized cannabis use, as well as the discrepancy between the states and the federal law is really challenging because in states like where we're located in California, there's a plethora of cannabis and cannabis products and cannabis consumption, but academics or federally regulated institutions and scientists have a really hard time and in most instances are forbidden from accessing that federally compliant, state compliant, federally illegal cannabis. I'll talk a little bit about why that is and some different options that researchers can pursue. Next slide, please. This is a slide that hopefully is a little bit of a stay behind as some of the members of the audience may want some resources later or to come back to the presentation. In order to understand the federal cannabis research landscape, we have to look back to 1970 when the Uniform Controlled Substances Act was passed. And that was a law that regulated controlled substances and really aligned US federal law with the United States' international treaty obligations. And what it did and what it still does today is that it codifies and legislates that the DEA, the Drug Enforcement Administration, the US's narcotics body, is responsible for regulating everything having to do with controlled substances. They describe it as a closed loop supply chain, meaning everyone that touches a controlled substance is very tightly regulated and monitored, not just the person or the institution, but the actual product itself, in this instance cannabis, is very tightly regulated. What that really means for the researcher or a member of the audience is that consumers can access state legal, federally legal cannabis, but institutions or academics cannot because they must procure their DEA approved cannabis research materials from a DEA licensed source. Historically, there's only been one of those sources, meaning a researcher can't simply go to a dispensary and purchase a product to study. They have to get it from a DEA approved source. Historically, there's been one, and the DEA will soon be approving several other applicants and manufacturers for cannabis research that can supply academics for partnership and collaboration. Next slide, please. So if you're a researcher and you want to access cannabis, what can you do? And I'm operating under the assumption during this part of the presentation that you choose to follow federal law. So I don't obviously say that you could break federal law and go purchase cannabis in a dispensary or from a cultivator. So your first option to access cannabis would be to go through the drug supply program, which is administered by NIDA, the National Institute on Drug Abuse, which is the only organization in the US today authorized to cultivate. That's the University of Mississippi. And they operate under a government contract from NIDA. The other option, and this one is a little bit challenging and intimidating if you're a researcher, but it's actually quite doable. And that is that you can import federally, legally produced cannabis from abroad. So if there's a place around the world, Canada, United Kingdom, Israel, some places in Latin America, and it's produced legally at their federal level, they can export that cannabis to you as a DEA approved researcher, what's known as a registrant here in the US. A lot of researchers don't know that. They just say, look, the University of Mississippi may not have the products and inventory that I need. I'm out of luck. But in fact, I'm here to tell you that you actually can go through the process to import products from abroad. The other thing you can do is, and this is very standard in the medical or the pharmaceutical industry, is that you can partner with another DEA or with a DEA registrant to conduct research on your behalf. That would be sort of the CRO or contract research organization model. We've done this quite a bit in the past successfully with institutions that perhaps that investigator doesn't have a DEA license or registration, but a DEA registrant could conduct that research on your behalf and then data share with you. So that's one way to work sort of within the system, again, assuming you're federal compliant. Next slide. So let's say you do want to become a DEA registrant. What do you need to do? As I mentioned at the very beginning of the presentation, it is challenging and cumbersome and can be quite intimidating, but it's well worth that path, and it's a well-trodden path, and it can be done. So don't be intimidated. This is a list of some of, and certainly it's not exhaustive, but some of the interagency groups that you would need to procure approvals from to become a DEA researcher. Obviously, the Drug Enforcement Administration, you need a Schedule I marijuana, they spell it with an H, marijuana registration for your research. If your products are going to go into a human, most likely, or if they're going into a human, you're going to need FDA approvals. You may end up needing NIDA approvals as well. And then one thing that a lot of researchers overlook is they say, look, I'm regulated by the Fed. I've got to go through potentially NIDA, FDA, DEA. Okay, I'm going to jump through all those hoops and I'm good. What oftentimes people overlook is that the DEA oftentimes considers itself the regulator of last resort, meaning you still have to have all of the local and state approvals to conduct that research. So here in California, we have to have approvals from our Department of Justice, what's called the Research Advisory Panel. And then naturally, your institution will almost certainly require IRB approvals. And your university may even require that you have approvals internal to your controlled substance research department. All that to say, engage early and often and over communicate with all the stakeholders, because it can be quite cumbersome, because you would think that this process flows sequentially and linearly. Step one, talk to DEA. Step two, talk to FDA. In fact, it's not, and it's not entirely uniform. So engage early and often with all of the organizations listed above. Next slide. So let's say you do proceed to go down the DEA registration path as a cannabis researcher. What do you need to do? Understanding a bit about what the DEA cares about for you as the researcher is quite important. The DEA's charter is to administer, maintain, and protect the closed loop supply chain of controlled substances. That can be what they call through their diversion control division, and then that can lead to enforcement actions, which is sort of what we more traditionally think about the Drug Enforcement Administration doing. The diversion control division is really going to be who's going to regulate you as the researcher through their local field office. So they're going to do the required inspections of your laboratory, your facility, your clinical site, if it may be on your campus, and they're going to do a lot of paperwork. It's complex, timely, and doable. As I said earlier, I very highly recommend if you go down this path, start as fast as possible, because it can take, I would say on the better end, 6 months. In fact, in some instances, it can take 12 to 18 months to go through this process. I'm not going to talk to the bullet points there, but as a takeaway, if audience members have questions about certain forms, et cetera, I'd encourage you to refer back to this slide. It can be, I will say, it can be quite challenging to navigate this process. We're happy to help partners, investigators, researchers. We're happy to help them navigate it. Next slide. A couple of concerns, and again, these are resources for you in the future, but once you've gone through this process to decide to become a researcher, there's several concerns that you should have about the cannabis study materials you're going to have. The first is quality. The FDA, if you're going to give the drug to a human in some of your research, is going to care about a lot of things that typically stem from the ability to ensure traceability, reproducibility, and that's all done through your supplier's pharmaceutical quality system, quality assurance, quality control, and their full-fledged quality management system. Again, I encourage you to reach out to a partner or a supplier as early as possible to make sure that their products are going to meet your needs. It would be a disaster to go through this lengthy regulatory process, line up your partnerships and collaborations, which AJ will speak to, and then all of a sudden you don't have an appropriate source of materials. So start thinking about this early. Next slide. One of the things that you should also care about as a researcher is the actual production environment of your materials. This will feed into the quality management system that I just spoke of. If you're going to do research on something like any other product, you want to ensure that it's it's high quality and it's produced in a consistent way whereby your inputs are known and standardized and characterized. Again, the quality and the output of your research is going to be dependent on the input, and ensuring a high level of production, high quality production from your potential supplier through some of the mechanisms I described earlier, import, NIDA, or working with a partner. That partner's sophistication of production and level of production is going to have a direct impact on the quality of your research. Next slide. Again, your partner should be able to provide you with a very high degree of confidence that the pharmaceutical, biotech, and mechanical industries are considered part and parcel. They should be able to provide you with an adequate level of comfort and documentation that the products and the quality of the products have been analyzed and verified, such that your research will reflect a high caliber of inputs and products. This is all done through analysis in a laboratory setting. Next slide, Chad. I'd like to share a couple vignettes just to wrap up and before turning it over to AJ, with just a couple things that some of the members of the audience sort of may be wondering. These are a couple places where we've been able to help some academic and research partners. And I imagine these two scenarios, perhaps some of the members of the audience have dealt with them before, perhaps even yourself. We were approached by a university and they said, hey, I'm a researcher. I've got the Schedule I material. I've got my Schedule I research license. They jumped through all the appropriate hoops, but at the end of the day, the NIDA program, through no fault of their own, didn't have the materials this investigator needed. So they'd gotten all their approvals, their funding. They got to the very key culminating event of actually receiving the test material and the study drug, and it wasn't in inventory. What do you do? And as some of the folks here may know, a lot of these approvals, the state and federal approvals, have timelines attached to them. So once you get your approvals, you're really under the gun, which speaks to the importance of getting all of these things lined up from the start and from scratch, really, to be able to execute on them. One of the things we did with this partner is we worked with some of our international partners that are federally compliant to assist in matchmaking and assist the investigator with partnering with an international company to import federally compliant study drugs. So it's quite a challenge, as you can imagine, to get the appropriate country export permit, the appropriate DEA import permit, and then finding someone effectively, not to simplify or to be crass about it, but to find a freight forwarder or customs broker to fly that cannabis material legally with the appropriate permitting to deliver that study drug from an international country to the actual university. Logistically, it's quite challenging. Now, we were able to get some really high THC flower to this investigator. We were really proud and happy to support them because they're doing fantastic work. So if that describes any of you, we're happy to talk about it later, how we can help. Another example is we were approached by a PI that needed to analyze some data, some analytical work around cannabis vapor. But this person didn't have a DEA Schedule I license. And they were concerned that they needed to execute their program as fast as possible, but they didn't have the time to wait for all the regulatory stuff. So we worked with them in sort of what I would characterize as a little bit of a CRO, contract research model, whereby we conducted the investigation with our federally compliant and partnered in the investigation with our federally compliant cannabis, and then allowed this investigator to access the actual data. And in that manner, the investigator got what they wanted, because they needed the data with which to conduct their study and analysis and publication, but it didn't require them to actually get hands on the cannabis plant. Again, just another vignette that I hope is possible, hopefully illustrates that industry is ready to work with academia, work with researchers in the research community. And I hope it speaks to the point that sort of, if there's a will, there's a way. And I think what most of the audience members know is there's an incredible need for compliant cannabis research right now for all sorts of indications, agronomic research, et cetera. So I encourage everyone that's attending to know that cannabis research can be done. It's challenging and it's hard and it seems intimidating, but if you start early and you engage often and you work with good partners that can help shepherd you and guide you through that process, your work will see the light of day, will be published, and certainly will be appreciated by those of us that are your peers in the community. With that, I will pause, Chad, and thanks everyone very much. I look forward to later in the session for the Q&A.
All right. Thank you so much. I certainly can speak from personal experience that there have been a lot of questions that have come to the ICR about conducting compliant cannabis research. And it's good to learn about the resources that are available and what can be done. If you've entered a question into the Q&A window, we are going to go ahead and hold off on answering questions until the end of both presentations. So please hold on to those. We will certainly get to them when we can. It's my pleasure to introduce Dr. A.J. Nayak as our second presenter this afternoon. Dr. Nayak is an assistant professor in the Department of Medicine at Thomas Jefferson University, Philadelphia. He has an appointment in the Center for Translational Medicine and the Division of Pulmonary, Allergy, and Clinical Care Medicine. Dr. Nayak received bachelor's and master's degrees in microbiology from the University of Mumbai and his doctorate degree from West Virginia University. Dr. Nayak has been actively involved in cannabis research efforts for almost a decade now. His primary areas of interest are in the field of allergy, but he is also interested in examining the physiological and pathophysiological effects of cannabis on human health. He is engaged in multiple collaborations, both nationally and internationally, to further cannabis research. His lab is active with a grant from NIH to develop diagnostic tools for determining cannabis allergies. And before I turn things over to Dr. Nayak to present, I think it is worth noting that he is with us today despite recently breaking his hand, which will require surgery, and is in the midst of moving. So we do appreciate you making the time to present today. With that, I'll turn it over to you.
Thank you very much, Chad. This is a great opportunity for me to share my experiences of working on cannabis. And really my goal here is to give a sense to researchers, young investigators out there who might be trying to go after a research question that they want to build careers on. And so I want to give a little bit of an insight into how I went about it on the topic of cannabis and how forming these partnerships and collaborations have actually helped my career. So I'm hoping to give some kind of detail around that. Aptly my title, the title of my talk is Navigating Through Cannabis Research, an Academic Story of Partnerships and Collaborations. So my research, my postdoctoral research started at the CDC campus in Morgantown, West Virginia, where they house the National Institute for Occupational Safety and Health. And my mentor at that time, Dr. Donald Beesholt, was approached by one of our friends and longtime collaborators from Canada, Dr. Gordon Sussman, who started seeing a lot of patients who claimed to have allergic reactions on exposure to cannabis. They were from different walks of life. Some were occupational exposures, some were recreational exposures. But nevertheless, we saw that there was some kind of reaction on exposure to the plant. We didn't really know much about it. We discussed about like if we wanted to go after this question, how would we pursue that? So my first step was to really look into the literature and understand, how, what is really known about this, right? So I found a paper, which was published in 1970s, which seems to be the first paper to describe some kind of an anaphylactoid response after smoking marijuana in an individual who claimed to smoke it for the first time. And then certain, laboratory tests suggested that it's possible that cannabinoids in the plant could potentially have triggered this reaction. Since then, we did come across a few additional papers. They're mostly case reports, really, describing recreational and occupational exposure to cannabis resulting in some kind of an allergy or allergy-like reaction. The symptoms are fairly broad, with dermal symptoms, with typical urticaria or hives on contact with the plant, or respiratory use resulted in like rhinoconjunctivitis, some lacrimation, fairly broad enough. But we kind of recognize that, as more and more of these case reports started to come, that there were different ways that one could potentially get exposed. That there was smoking, which seemed to be the primary route, was the one driver of the upper airway and lower airway symptomology. Contact which is probably an obvious interaction with the plant prior to smoking, or even in the occupational setting where somebody might be touching it directly, was also a route of exposure. In rare cases, we found that individuals who had ingested cannabis as a tea or some kind of an edible also sometimes, showed reactions of mostly gastrointestinal nature. And there were a couple of reports that talked about the potential exposure to pollen and how cannabis pollen could drive this kind of reaction. Most of these reactions are fairly mild to moderate, with very rare scenarios of anaphylaxis. So when Dr. Sussman's group came to us and they talked about these patients, like, how are we going to go after this question? If it's really a cannabinoid, then that would have been very challenging to really study, mostly because we would have to seek all the DEA clearances. And even if we did, then we would have to develop assays, which would take a long time. Nevertheless, there was a simpler question that we could have gone after, which was, we approached it in a manner that, okay, cannabis is a plant, just like people are allergic to plants of different kinds, pollen. Is it possible that you have patients developing IgE antibodies, which drive the type 1 hypersensitivity to some kind of proteins from these cannabis? And what are these major cannabis allergens that elicit this kind of a response? And if that's the case, then can we develop a diagnostic sort of measure to allow for definite determination of allergic sensitization to cannabis? This especially becomes important because cannabis is a very complex, entity. It has so many different components, and you do not really know how these components are interacting with each other. But by focusing it entirely on the proteins, we removed sort of any possibility of interactions from other components. Now, in clinic, what Dr. Sussman was able to do is that when he had patients presenting with potential symptoms from exposure to cannabis, he was able to grind up some of the material that they were using and then make like a saline solution with the material and test it on the skin, where they inject it into the skin and then see if they break out into a wheal and flare reaction, which is typically identified as skin prick testing. And there was some variability between the patients, but nevertheless, all of them demonstrated a positive reaction over the controls used in this study. Now, as you look on the slide, most of the exposure was through smoking or through contact exposure. And urticaria and conjunctivitis were the very common symptoms associated with this. But what we didn't see was really a set of individuals who had consistent other allergy reactions. Many of them had prior allergies, but they were to different, organic sources. Some to dust mites, some to fungi, some to other pollen, peanut allergies, drug allergies. And what struck us was there were quite a few individuals who had never seen any other allergic reactions. They were not known to have any other allergic reactions. So it was a very diverse sort of a patient profile. Now, in order to examine what truly are, what cannabis proteins are these patients reacting to, we had to test this in the laboratory. So Dr. Sussman gave us the serum samples that were necessary to conduct this investigation, but we needed the plant material. Now, we recognized that bringing in cannabis plants and trying to macerate them and make extracts in the lab was out of the question. So when we tried to go after this question, we recognized that University of Mississippi does grow cannabis. It's a federally supported program. And interestingly enough, we also were educated by the fact that while cannabis in toto is sort of a restricted substance, cannabis proteins and the genetic material are actually not under the Schedule I limitations. So this was kind of helpful because then we could get protein extracts from the plant shipped to us for analysis. And this was really very helpful because when we ran these protein samples on a gel matrix and then transferred it onto a nitrocellulose membrane, which allows the binding of proteins, and when we probed these with patient sera, we noticed some patterns, right? So those individuals that were skin prick test negative but came into the clinic with some kind of symptoms to cannabis, we noticed that their overall immunoreactivity to cannabis proteins was kind of weak. But we found that amongst those individuals who were positive, there was some degree of consistent pattern in immunoreactivity to different proteins. So we see a few different proteins here at about 20 kilodalton and maybe even like at 50 kilodalton, right? So we saw about half the patients that had positive reactions were demonstrating some immunoreactivity. To further understand or identify rather determine the identity of these proteins, we ran them on a 2D gel electrophoresis, which allows for separation of proteins based on their isoelectric point, and then the second dimension being the molecular weight. And we identified a lot of immunoreactive points that we were able to remove and then look at with mass spectrometry. And we identified at least a couple of different allergens, like rubisco, which is one of the most abundant proteins in nature because it's so central to the photosynthetic process. And another photosynthesis associated protein called the oxygen evolving enhancer protein. We did find hits for some other proteins, but they were fairly minimal. Now, what this did is that this set us up for one paper where we published it, and we determined that there's a possibility that patients, at least some of them, have IgE in circulation that reacts with cannabis proteins and potentially drives the allergic reaction. Now, at the same time, across the Atlantic, out in Belgium, Dr. Didier Ibo's group was also doing research on cannabis allergies. However, their sort of approach to this was a little different because most of their cannabis allergy patients also had a lot of food allergies. And so they published these studies where they recognized that the serum IgE levels to cannabis extract, when tested with cannabis extract, was much higher in patients who had cannabis allergies and plant food allergies relative to those that only had plant food allergies. And there was some sort of a functional significance to this investigation because they were able to isolate basophils and then stimulate them with these cannabis extracts and determine that the individuals with dual allergies to cannabis and plant food allergies demonstrated a more robust response in basophil activation relative to the only plant food allergies. And then, other investigations out in Spain also showed that similar to our investigation, there were multiple cannabis proteins to which individuals may have IgE. And in their investigations, they came across this non-specific lipid transfer protein, another allergen which was speculated as a profilin allergen, and a thaumatic-like protein. And the basis really was that some of these allergens that they identified here have sequence similarities to other proteins from other food sources. So up to 2016, this was the list of allergens that had collectively sort of come out, CANS3 being the only allergen that was registered with the IUIS nomenclature. And then the other allergens were out there in terms of like some level of investigation, but really the validation was still lacking. Now, in the last few years, what we've seen is that profilin has been validated and now is identified as CANS2, the oxygen evolving enhancer protein that we reported in our study in the US was also validated in Belgium and is now identified as CANS4. And more recently, another allergen, CANS5, PPR10 homolog, was identified. So this whole story about, you know, potential IgE to cannabis protein seemed real. So what was so interesting about the non-specific lipid transfer protein is that it came as a very strong hit in most of the European studies. And while we recognize that, one thing was very apparent to us is that in Europe, the CANS3 or the non-specific lipid transfer protein is the relevant allergen. But at least in the limited investigations in North America, mostly through our manuscript, we found that Rubisco was probably the major allergen, or the OEEP2 was the major allergen, but not CANS3. So, you know, we started looking at the basis of the lipid transfer, mediated cross reactivities. So we did a sequence alignment, and we found that the protein has very high degree of similarity to other non-specific lipid transfer proteins from other sources like walnut, peaches, peanut, hazelnut, tomatoes, mugwort. So, and these sequences suggest to us that it's possible that any kind of IgE binding to a certain set of sequences could also be preserved when it comes from another source, as seen here. We identified that there are about 8 conserved cysteine residues that help keep this protein in a certain conformation. And we also recognize that these are resistant to protease activity, meaning they're very highly stable allergens, which contributes to their potency. And as I've already discussed, because of this distribution of the lipid transfer proteins being so ubiquitous, in Europe, it's now very clear that a majority of their population that reacts to cannabis has some kind of a cannabis fruit vegetable syndrome. But this has not been extensively investigated in the United States. The one interesting set of studies that did come from Belgium was that as they were going after this non-specific lipid transfer protein as being a major allergen, they recognized that it may not be as relevant to occupational exposures or occupational allergies to cannabis. So in this one study, they tested serum samples from multiple law enforcement officers who had talked about potential allergic sort of reaction on exposure to cannabis, whether it was when they were doing it as part of their law enforcement activity or as part of like forensic activity. And only one responder really showed positive reaction on stimulation with this non-specific lipid transfer protein, suggesting that that the context of exposure is so relevant to really understanding what allergens are important. Now, because of this question of occupational exposures becoming relevant, and with my postdoctoral fellowship at the National Institute for Occupational Safety and Health, we started engaging with other folks within our group who are interested in this question, which got us to involve with NIOSH group out in Ohio and the Western State Division out in Colorado. And the Colorado group was actually very important because Colorado was one of the first 2 states to give access to cannabis, and from our standpoint, potentially give us access to more patients who'd come from these states, and access to the serum sample. So then this was the next sort of link in this collaborative network. Now, I don't want to go into the details of all these studies, but in the last 3-4 years, we've published multiple papers where when I was part of NIOSH, we went to, I had the opportunity to visit a cannabis grow farm in state of Washington, and we look at and we conducted a hazard, HHE evaluation, where we looked at the hazards that are prevalent in those kind of environments. And these studies are published, they're available out there. What we recognized is that there are other things that could potentially influence our ability to understand allergic sensitization to cannabis. For example, mold is a very common sort of thing that is associated with cannabis plants, and mold can cause allergies too. So this gave us, introduction into a new nuance, really, that mold as a contaminant could also interfere with our ability to understand and interpret clinical, allergic sensitization to cannabis. Now, in the last few years, what we have done is, because of this surge in really these reports of allergic sensitization to cannabis and multiple people really getting involved, and thanks to Dr. Bill Silvers from University of Colorado, who's been able to get everybody who's interested in this research question together, that now this collaboration spans not only through multiple states within the United States, but also involves partners from other, from especially Europe. And now we have a large group of individuals who basically want to further the education and the knowledge and the research when it comes to cannabis allergies. This all came from us, just starting with one question, really, about, is allergic sensitization to cannabis really possible? And now we have gone to the point where we recognize its complexity, and really being able to do this in a collaborative manner helps get to where you want to be faster. But there are limitations and challenges, and I just want to point out a few of them. This was from Dr. Silver's study a few years ago, where what stood out to me really was the variability in the individuals who demonstrate reactions to cannabis, right? One patient smoked it frequently. The other had never had any history of allergies or asthma. And then we had this, it also had this component of working at a dispensary or a marijuana growth facility. And the third one being where the individual was a chronic user, right? So it's hard to really pinpoint on who potentially could develop an allergic reaction, whether it's a naive person or somebody who's got circulating antibodies. It's a very complex sort of thing. This is why we need to develop better diagnostic tools to help people. One challenge that I have noticed is that a lot of the work that has been done to date has been with cannabis sativa as the source of the research material. But we recognize that it's not really that straightforward. Cannabis indica is another major strain. And then there are a lot of hybrids of these two strains that are also available. While practically it becomes a challenge to really investigate this, we have tried to now focus more on the chemotypes, the high THC or the high CBD, or sort of like a mixed strain, to keep it at least within the realms of what's practical in terms of our investigation. This line that I don't remember where I came across, but it said plural of anecdote is not evidence. A lot of the research that up to date has sort of been published has focused on these individual case reports with significant variability in the research material used in the lab for the research studies, or the material that's used on the patients too, because there's really no standardization for this. In some cases, patients are not willing to disclose or are not able to recall symptoms. The strain variability, as I discussed earlier, is a major challenge. And the fact that, getting to the point of the standardized reagents, which is essential for diagnostic tool development, there are a lot of hurdles to overcome there as well. What we have started doing is really that rather than go after the plant, because the DNA or material is available, the genetic material is available, the RNA is available. A lot of the focus now within this collaborative network has been to develop recombinant proteins for different allergens or potential allergens, and then use them as the material for diagnostic testing and standardizing diagnostic testing. So this conversation has moved further along because now we have sort of become more focused on individual components, which allows us a lot more room to experiment and validate different targets. I think that a lot of whatever I have read about cannabis, it's kind of captured by this one phrase by the Humboldt County supervisor, who in some article he said this, or he or she said this, Humboldt County being a site where there is a lot of cannabis that's grown, and they saying that we are building an airplane as we're flying it. And that's how I feel sometimes with cannabis, is like that. Now, if you look at the landscape when it comes to cannabis legalization, it's changed a lot significantly from 10, 12 years ago, with Colorado and Washington being the first two states to increase the access to cannabis. There are more states now talking about legalizing it, decriminalizing it. That's why I have asterisks against all those categories. And so whatever our sort of thinking is about this, this is happening. So it is important for us to get to the point where we are armed with the tools that are necessary to really study the impact of cannabis from both physiological standpoint as well as pathophysiological standpoint. And so this is kind of important. Most of my current research is sort of furthering this development of diagnostic tools, and we've now have industry partners as well who are helping us develop these sort of assays. And I think that with North America specifically, we've been sort of interested in sort of going after, is LTP really a relevant allergen? And some of our preliminary data suggests that it is, but again, the context in which it is needs more investigation. We are also committed now to trying to understand the allergens that are relevant from an occupational setting as well. And these are kind of like, the objectives of this larger collaborative research, and that now involves over 40 individuals. With that, I want to thank my lab that has been working relentlessly through COVID-2 in generating data. The support of my old mentor, Dr. Donald Beeshold at CDC NIOSH, that has, now we have a mechanism by which we can actually study allergic reactions in occupational settings in Colorado. Dr. Gordon Sussman, who was the first one to come up with this question, and then supporters at University of Mississippi who provided us the abstract. At University of Antwerp, Dr. Didier Ibo, who's published immensely on this and has been a great collaborator. And then, Dr. Ellen Burnham, with whom we recently wrote a grant. It looks like it's going to get funded. She's been very supportive of the research questions that my lab has come across. And Dr. Bill Silvers, every field needs a champion, and the field of allergic reactions to cannabis, Dr. Bill Silvers is the champion who's allowed us to be able to get in this position where we've been able to secure grants and actually make progress. And then finally, I want to thank the Lambert Center at Thomas Jefferson University that has supported us in many ways to really further our interest in sort of, our commitment to improving the knowledge and research on cannabis. Thank you.
Thank you so much, Dr. Nayak. Fascinating work. We have had a number of questions come in, so I'm going to go ahead and jump into them. We'll see if we can get through most of them before we reach the end of the hour. So it looks like this first one would be primarily for Mr. Hodgin. How long is the process for cannabis import permitting what are the biggest challenges with importing? And are there specialized carriers, or is this similar to FedEx Cannabis with its shipping via UPS? Or I'm sorry, similar to Federal Cannabis with its shipping via UPS.
Great. I will, there were 3 questions there. So Chad, if I missed one, please ping me or suggest. So the first sort of, what is the process like? The recipient, in other words, the importer, has to be a DEA registrant. So you still have to be a Schedule I registrant. The process goes something like this. You identify the party, counterpart, company, whatever, who's going to do your export. Then the importer would contact the DEA import-export section. You get a DEA import permit. Frankly, that's a lot easier than, or it's a lot more well-trodden of a pathway than you may imagine. It does not take very long, like typically less than, call it 2 weeks, can be 3, can be 1, to get your actual importation permit. Then you would send that material to the exporter. The exporter, and there's a little bit of nuance, but in the interest of simplicity, the exporter would then get an export permit from their country. For example, if you're exporting from Canada, you would get a Health Canada export permit. And you have to do those sequentially because they effectively get married. Your export permit marries your import permit. And at that point, you're sort of cleared to engage. And at that point, the timeline and the process can take several weeks. I would handicap it, depending on the country, 1 to 2 weeks, can be 3. And that really is a customs clearance process, because unlike just drop shipping something with FedEx, you know, it's declared and it's a controlled substance. So it's going to go through Customs and Border Patrol inspection. There's a clearance process involved. And that's where you need a good partner, either an exporter. We're, again, happy to help with stuff like this, to identify the appropriate customs clearance agent or freight forwarding company that will actually handle the transportation door to door. So they're going to go pick up, it sounds simplistic, but they're going to go pick up the cannabis in their delivery van from your exporter, take it to the airport. They're going to meet it at the airport and make sure it gets walked through customs, and they're going to drive it right to your laboratory. So it's a lot of steps logistically, but it's not as challenging as getting that DEA registration. That's the real key. And then the second real key is having a good partner with whom you have trusted freight forwarders and customs clearance to really kind of hand walk the product through the process.
Okay, thank you. I think the next one, this looks like it's appropriate for Dr. Nyack. How has the route of exposure, or I think you used the term occupational exposure, influenced the needs of your research team with respect to cannabis products?
Can you please repeat that question?
Sure. How has the route of exposure influenced the needs of your research team with respect to cannabis products? So with respect to the cannabis products that are needed for your research.
It's a good question. I think that what we have tried to do really is restrict our investigations towards understanding, you know, whether it's the occupational exposure or recreational exposure. Is there any evidence of circulating IgE antibodies to cannabis proteins? So from our needs perspective, we have mostly sought to investigate with cannabis extracts, protein extracts. Now, we don't really have much in terms of choice about which strain we do, which, you know, it's whatever is available to us, and we try to keep it consistent between studies for interpretation. So we just try to seek out those. Now, with occupational exposures, I think that the one context really is the plant that you're investigating, or the type that you're investigating, is it really relevant to occupational exposure? We're kind of getting only into it now. And there are different strategies around it. But at the end of the day, it's all going to go down to the plant. Now, we can break it down to the buds, you know, the leaves, the roots. Those are the different components, but that's pretty much it.
Okay, thank you. A question about BRC. So does BRC perform their own research on cannabis, or is it all through partnerships? If yes, what cannabis research are you performing, and how is it funded?
Sure. Let's see, several questions there too. So short answer is yes, we are performing our own research here. Largely, it's agronomic research. So we're not a clinical trial site. We're not at a hospital, so we're not administering the study drug, at our building, in a clinical setting. But we do a lot of our own analysis and research here. We're a private company, so it's funded through our company. We also do work quite a bit extensively with partners to do research, sponsored, industry-sponsored research, where a partner could do something on our behalf, and that's both sort of on the agronomy side and on the clinical side. We have a partnership with a university here in California, University of California, Davis, where they're doing a lot of the foundational plant science work in partnership with us to really understand a lot of the basic chemistry, biology that's largely been done for every other crop, if it's a strawberry or an olive or whatever. But on the cannabis side, a lot of the foundational biology and chemistry and analysis that goes into understanding plants, breeding plants, some of the things that Dr. Nayak was talking about, like how do these plants actually interact with the human or an animal? A lot of that basic work really hasn't been done to the rigor that is expected in the research community. And so we're working with them to conduct a lot of that agronomic research.
Okay, great. This is going to be the final question as we're nearing the end of the hour, and this one is for Dr. Nayak. Do you anticipate that more people will develop an allergy to cannabis given the increase in cannabis use in the past few years?
I think so. I think it's a simple sort of a, I don't know how prevalent it will become. Most of the reactions are mild, reactions to it. I do anticipate that as it becomes more accessible, especially in the recreational space, I would say, that we potentially would start seeing reports of this. I think a lot of the reasons why people are not coming forward yet because of possible, prosecution or, other fears, right? So with, for example, in Canada, as soon as it was made legal is when Dr. Sussman started seeing a lot more of these cases. Is it going to be like a major allergen? I don't think so. At least, the current evidence doesn't really support that, but it is going to increase. It's going to become, more commonly reported. I just can't comment on how prevalent it's going to be.
Okay. Well, thank you. I want to thank both Mr. Hodgin and Dr. Nayak for their time today and the information that they shared. We appreciate the time and effort that goes into that. This concludes the webinar for the month of May. I hope you will join us in June as we will hear from Dr. Clayton Schnell from Greenwich Biosciences, who will be taking us into a deep dive into Epidiolex. Have a good afternoon.
Thank you, guys.
Thank you.